Our director, Dr. Beryl Morris, sends her apologies for not being able to host today. She is, however, listening to the seminar while in transit. Before we start this webinar, we acknowledge the traditional owners of the lands where UQ now stands and recognise that these have always been places of teaching and learning about land, water and culture. We also acknowledge the traditional owners where each of us are located today and pay respect to elders past, present and emerging, and recognise the continuing important role of Aboriginal and Torres Strait Islander people within our community. I'm located on the land of the Kaurna people of the Adelaide Plains, and their traditional welcome is Nina Mani. Hello, how are you? So on to some practicalities of the webinar today. We can't see or hear any of you as attendees, but you can submit questions online. At the bottom of your Zoom window, you'll see a Q&A button. And if you click on that, you can type in a question at any point during the seminar, and we'll be answering those questions at the end. Today's webinar will also be recorded so that we can share it later with those who couldn't be with us now. This is the second in a series of webinars that we commenced this year, and we hope to continue as a regular event. Most of you will know TERN partners receive funding from the Commonwealth Government under the National Collaborative Research Infrastructure Scheme, and that enables the 3 platforms of TERN to operate across the continent. Today's talk sits within the Ecosystem Processes platform, and you can see that our partners and co-investors are shown on the slide. We have 2 presenters today, and the first is Will Edwards. Will's an Associate Professor of Ecology and Zoology in the College of Science and Engineering at James Cook University and Program Leader of the TERN Ecosystem Processes platform. His research investigates a wide range of ecological patterns and processes, and his work is very much cross-disciplinary. He is involved in projects as diverse as the use of entomopathogenic fungi to control fruit fly pests, understanding carbon cycling in tropical rainforests, evaluating hunting sustainability and the potential role of protected areas in the use conservation area of Papua New Guinea, and the impacts of acoustic properties of introduced species. Our second speaker is Joe Owens, a systems modeller and researcher with over 20 years experience in water balance and eco-hydrological modelling. Joe is a research fellow at the Centre for Agricultural Engineering at the University of Southern Queensland and works closely with the Queensland Department of Environment and Science on joint projects funded by the Drought and Climate Adaptation Program and the Queensland Reef Water Quality Program. She has extensive experience in modelling at the landscape, catchment and regional scales for runoff, soil erosion, water quality, land management and evapotranspiration. She has a passion for parameter estimation, model optimization, programming with Python, and for working on collaborative projects across disciplines. I would like to now hand over to Will to get things going.
Hi, thanks Angela, and just while I share my screen, I hope everyone can see that. I'd like to just take the opportunity to thank everybody for taking time out of their schedule to come today. My name is Will Edwards. I'm not sure how many of you will know me, probably not too many, but I am the head, as Angela says, or the lead of ecosystem processes. And so I thought I would take a bit of time just at the beginning to make sure everyone understood what ecosystem processes was and where it fitted in within the TERN sort of framework. It's one of the platforms that gathers data, and it's the platform that is essentially at the sort of highest resolution or the smallest sort of area. It's done through we use intensive sort of field stations which combine both instruments and sensor measurements, which are the detailed, intense time intensive or time highly resolved temporal scales with sort of classical field surveys and remote sensing. And so where we sit is questions about why. And on the graphic on the screen, I guess we are the lowest, the blue part of that, what might you call it, a cone-shaped element figure, but we have cross contribution across all the different scales. There are currently 15 sites, and 12 of those are TERN funded, plus 3 affiliate sites. And so they are spread across the country in a comprehensive sort of, an attempt to capture a great diversity of different land types and land systems. And each of the super sites comprises sort of a main field site, which takes vegetative, faunal and biophysical measures. And so the core research, I guess you might say, at a super site would be the meteorological flux stations, plus plant physiological measurements, as well as long-term flora and vegetation monitoring. What we deliver, I guess, is micrometeorological observations of energy, carbon and water, as well as biological and environmental observations of the flora, fauna, microbial biodiversity, soils and hydrology. And to, I guess, contextualize the work, it's recognized both internationally, both as a major player in FluxNet, which for those of you who don't know, but I suspect many of you do, it's the global network of eddy covariance micrometeorological stations, and as well as sitting inside ILTER, which is the global network of long-term ecological research stations. I'm going to show you next a sort of graphic that has an awful lot of parameters, you might say, or data streams and data sets. And it's broken into 2 different sections. So the top half are the types of measurements that are associated or generated through the micrometeorological stuff. And they're continuous to half-hourly to annual estimates of the sinks and sources of greenhouse gases and water and carbon. And they're used for, carbon accounting and water management. And then the second, or the lower panel, is essentially the more traditional biological biodiversity types, which are done at, say, 6 months to annual to 5 years. And so their estimates of biological processes like vegetation species changes or compositional changes, leaf area index and ground cover and things like that. And so the lower panel, I suppose, is what you might think of understanding biological responses, whereas the upper panel is more the biophysical or micrometeorological responses. The types of applications that we use this information for are to things like the effects of disturbance, topography, stand age on, as you might expect, carbon and water fluxes, or to biodiversity. But it also, and this is the link to the landscapes element, produces important ground truth data for parameterizing and validating satellite remote sensing and global sort of inversion of carbon products. Some of the questions that we can answer, or that are answered with our data, are things like, what are the key drivers of ecosystem productivity? How resilient is that productivity and biodiversity to a variable and changing climate? And what is the current water budget for the dominant Australian ecosystems, and how might that change into the future? Some of the examples, the concrete examples of use, things like drought research. So we have our sites embedded into a global experiment that investigates ecosystem responses to drought. As I said before, many of our sites are recognized as highly valuable in the calibration and validation of satellite-derived products. And some of our long-term, especially, and this is particularly relevant in Queensland, the long-term biodiversity monitoring that's been done, say, in the wet tropics, provides a long data, a long time, I guess, assessment of how climate change has altered species distributions and species range sizes, or in the wet tropics at least. For Queensland, there have been there are four SuperSites. Now, it has history because, believe it or not, the very first two ever SuperSites that were sort of identified or defined were in Queensland, and they are the Daintree, which is DRO, and the Sanford peri-urban one. There are two others in Queensland now, which are called Robson Creek and Mitchell Grass Rangelands. And as a quirk of history, I guess, the two oldest ones are no longer the main funded sites from TERN. They are the affiliate sites. So remember I said that there are 15 affiliate sites. I mean, 3 affiliate sites across the country. Well, 2 of them are Daintree and Sanford. And so the Queensland government and TERN's RICF grant or co-contribution fund was aligned with things to bring back online and to increase the scope of the affiliate sites as opposed to the already funded TERN sites of Robson Creek. So in terms of Queensland, we have 4 individual sites, and each of those sites, while they do a core set of recordings and measurements, as I showed you before, they're in different locations with different histories and different vegetation types, and therefore they also answer different types of questions, or they're capable of addressing different processes or different questions. And I'll just run through them all in case you don't, some of you might not know exactly all of them. So the first one is the Sanford peri-urban one, and it's located in the Sanford Valley, about 20 kilometers northwest of Brisbane. And it's because it's located in a semi-urban or a peri-urban fringing sort of region, then its focus largely is on things like ecosystem services in the face of urbanization, so that it measures things like nutrient fluxes and catchment and downstream effects of urbanization. Essentially is a function of its position in the environment. The second original one is the Daintree rainforest, or it's sometimes called DRO, and it was also identified in the RICF as for instrument refurbishment, I guess. But as I'm going to show you later, a change in circumstances has left opportunity for something that I think is much more powerful and much more what might you say, much more sort of insightful, I guess, for us to alter our original ideas about that investment. The Daintree rainforest is no longer going to be instrumented for flux, but it will remain as a sort of affiliate super site in the sense that JCU produces or provides the 5-yearly and annual estimates of vegetation and woody debris and plant species identifications and stuff. It will just not have the flux material. The 2 that are currently funded by TERN, there's the Mitchell Grass Rangelands, which is out at Longreach in Western Queensland, and it, because it's a grazing land, has principal sort of concentration or principal questions that are associated with stocking density and the effects of stocking densities on net primary productivity and carbon exchange and evapotranspiration. And finally, the 4th one, which is the Robson Creek one. Many people from the Queensland government might recognise the Robson Creek one as being the associated with the 25-hectare plot, which was developed along the lines of the Smithsonian CTFS network to measure long-term change in rainforests. It's located on the Atherton Tablelands, and it is essentially also, like the DRO, principally aligned with identifying changes in biodiversity patterns and species relative abundances. So there are the 4 different sites, and I just quickly want to return back to this figure that I showed you before, because it separated the 2 sort of panels into something that we measure very at a high frequency, and some things that we measure at a relatively low frequency, or every 6 months or every year or every 5 years. One of the beauties, I guess, of the new instrumentation that's coming on board is that we begin to align the temporal resolution of the measurements that we take, both for the biodiversity and the meteorological stuff. And so I'm going to tell you about 2 different new things that are going out to all the SuperSites and beyond, in that going to, I hope, bring about a much greater connection between those 2 sets of measures, as well as revolutionize how some environmental monitoring is being done. It's TERN's goal, and TERN are working towards creating an autonomous sort of wireless sensor network, and that includes remote camera taps, and they operate alongside time-lapse vegetation cameras and acoustic monitors and climate sensors. And so we are, the beauty, I guess, of the autonomous wireless network is that it's going to allow us to build a much more complete picture of biodiversity and provide early detection of environmental change. The first is the TERN's involvement with A2O, and for many people probably understand that the Australian Acoustic Observatory is located at QUT, and a fellow called David Tucker is involved in both the flora and fauna monitoring at Sanford and Mitchell, but is deeply embedded into, or is attached to the Australian Acoustic Observatory. That, I don't know if you were all driving home while Amanda Vanstone was on the radio yesterday, but she had a little segment about the A2O network. What it is it's a continental scale acoustic sensor network that is going to be put out for 5 years at least, in a range of locations across the country. All of the TERN SuperSites, as well as a whole range of other sites, will have these, up to 90, I think, different locations. They are already established at both Sanford and Mitchell, as well as a number of different other TERN sites, but they will be rolled out across the entire thing. The things that they allow are a range of outputs that give you an estimate of biological activity at a much finer temporal resolution than we could possibly ever have. And so we can reveal both short-term as well as long-term change. You can potentially identify changes in taxonomic groups or relative abundances, highlight seasonal breeding activity, and a range of other things. So it has the possibility of really revolutionizing what we know and what we can detect in terms of biological change, or biodiversity change over time. The second component that we're just about on the verge of deploying to all of the sites, so we'll go to all the Queensland and all the TERN EP sites, is the real-time imaging for plant performance. So we have these phenocams that are about to be rolled out to all of our sites, which will take a daily record of the colour of fields of interest, or field areas of interest, of which you can return estimates of plant performance, such as the green chromatic coordinates and so on. Now that's essentially all of the elements of the Queensland current SuperSites as they stand. But I did say at the beginning that I wanted to return to the Daintree one because the Daintree original bid for the Queensland Government and TERN Infrastructure Collaboration Fund was to re-instrument the Daintree Rainforest Observatory Tower. The image on your right is the rainout shelter that's part of the drought experiment that is currently underneath the tower at the Daintree Rainforest Observatory. Now that essentially means that it's probably not a great idea to re-instrument the flux tower measurements, given that we know in advance that this is going to influence what those measurements are. The beauty of that is that it allows us an opportunity for no extra cost, so a completely cost neutral, to realign or rethink about where we were going to put those instruments. And I think it presents a really interesting and really important alternative to the re-instrumentation of the DRO. And that is the reconsideration of the instrumentation moving to a Fletcherview station. Now, Fletcherview station is vastly different from the DRO, I'll grant you that. It's essentially a savanna woodland, just a bit northwest of Charters Towers. It's a operational cattle station that is owned by JCU. It's very close to the Burdekin River, and there's a whole range of historic information which Joe will allude to or tell you about, that makes this a very, very promising location to re-change or to center the focus of this monitoring. There are a couple of reasons why it's really important. First of all, it represents environments that are currently not well surveyed through TERN. So this on the figure here shows, and you can't see it terribly well, but the red dots are the surveillance, so the individual biodiversity monitoring plots from the surveillance platform across Queensland. And as you can see, there's virtually none in the location around where the proposal will be. So this will give us benefit. It's in areas that represent environments that are likely to change. So this shows how there's going to be the Hell's Gate Dam and Big Rocks Weir, and providing baseline information about those prior to their change is important. And then finally, it aligns with TERN Biodiversity Initiative. So there's another initiative similar to the Queensland initiative in WA, where we're proposing transects of biodiversity monitoring plots across areas expected to demonstrate a big climate change response. I probably should stop there. I think I've gone way too long. I'm sorry if I have. I'll hand over to Jo, who will discuss her work with using the data from flux towers and satellite-derived data to improve evaporation and evapotranspiration and ground cover modeling.
Thanks very much for that, Will. I'm really passionate about this topic, so I really appreciate the opportunity from TERN and the Queensland Government to talk about our work. Flux towers and water fluxes are my most favourite thing to talk about, so I'm really excited to be part of this collaboration with the new flux tower in Queensland that Will just introduced. But before I talk about the new flux tower, I'll talk about how we use TERN data in our work in combination with remote sensing and other data sets to enhance our modelling in Queensland. So we've come full circle with the energy balance modelling in our group. Many of the people in the webinar today, particularly those from Queensland, will recognise Greg McKeon. So our story starts with Greg when he collected experimental data at Catherine and built his first pasture and surface energy model, which later morphed into the models that we use operationally in Queensland today, the GRASP and Aussie Grass suite of models. And I started my career working with Greg McKeon many years ago in Brisbane when we used to be at Indooroopilly. And I feel like I've come back full circle as well, back into the group that Greg started. And so this is the group that I work for now. So hopefully many of you recognize the Silo Climate Database, where a lot of researchers and modelers get their climate data from. The GRASP model is disseminated through this platform as well, through Forage, which sits here, and Aussie Grass that sits here. So Aussie Grass is the 5-kilometre continental scale version of GRASP that is led by John Carter, and John actually leads all the GRASP modelling in Queensland and Australia at the moment. And Forage is our other product. So Forage is a whole of Queensland scale, and it's run at a property scale, and anyone can submit a Latin long or a lot plan number and get pasture growth estimates and stocking rates for any property in Queensland. And my role in this group is in the engine room. So I work in the model development and improvement and testing of the GRASP model, the core component of GRASP that gets used by all the other versions of GRASP. So one of the things we do is continual improvement with new data sets. And we found lately that TERN seems to be a one-stop shop where we get a lot of our data from. So we use the soil and landscape grid of Australia data that sits under our models. We use the AusCover and the remote sensing data, AusFlux, and also the Cosmos soil moisture data. So this is the reason why we started doing this work and how I first started using the flux tower data. One of the areas that needed improving in our model was evapotranspiration and the water balance. And evapotranspiration is such a large chunk of the water balance, and we knew that we were overestimating evapotranspiration on a daily level, especially in areas, especially on events straight after a rainfall event. And so we knew that that needed fixing, and we found the flux tower data to help us with that work. But before I do that, I'll just also let you know why this work is important. And so the GRASP Forage and Aussie Grass is used operationally in Queensland for drought declarations. The Queensland Rural Fire Service uses it as well, and the Department of Agriculture, Extension and producers use this to work out how much pasture is available for graining sustainably over the growing season, and also when to destock. And so that was the main reason why we started improving this. And the other reason for improving this is we noticed that the areas where we were having trouble was areas in Queensland that had woody vegetation. We've got about 100 sites all over Queensland where we measure soil moisture and pasture growth, but these are all mostly sites that are grasslands without any woody vegetation. And so 80% of Queensland is classified as grazing lands, and half of that has woody vegetation. So there was a large chunk of Queensland that we needed to improve this modelling for. This is just a conceptual model of GRASP. It's essentially a soil water balance, pasture growth and vegetation model, and we also model grazing management. So in addition to to modeling ET, pasture growth and animal production, one of the things we wanted to do with this particular piece of work with the flux tower data was to make sure that we not only got the total evapotranspiration right, but we wanted to try and get a better estimation of the various components of the partitioning, such as the soil evaporation, tree transpiration, canopy evaporation, grass transpiration, and and before we did that, even trying to get the runoff right and the deep drainage and recharge roughly right as well. So I'll show an example of where we did the first bit of work. That was at one of the TERN SuperSites called Alice Springs Mulga, which is one of Jamie Cleverly's sites. And so the reason why we did that was a large chunk of Queensland is in the Mulga bioregion, and also there's a fair bit of Mulga across Australia. So about 20% of the continent is under Mulga savanna woodlands, and so we thought we'll try and get a good calibration for those parameters. So this is, for those of you who haven't seen a flux tower site before, this is what the Alice Mulga looks like. So it's Mulga woodland, and we've got daily evapotranspiration data from 2010 to present. The other data set that was very valuable to fuse together was a time series of remote sensing data of the tree canopy for the foliage projected cover, and also of the grass understory, which is the fractional green cover. And that just helps us with the greenness to get the dynamics of pasture growth. And so the reason why we use the fractional ground cover rather than the NDVI was that the fractional cover is a biophysical measurement. It's actually been ground truthed against measured data. And so we found that it was a much better data set to use to directly compare against the grass model output. And things like green cover and total cover is really important for our pasture modeling, and it's also really important for our partners working in the paddock to reef, because cover is really important for degradation, runoff, erosion modeling. I'll skip over the long story about all the model improvements and the model optimization work that we did and skip straight to the results. But before I get there, I'll just show you this one slide. So when we started doing this work, usually when we model grass species in Queensland, we tend to parameterize the different grass species separately based on the water use and the growing habits of each of the grasses. And we thought we might have to do the same for the tree species as well. But we found, when we found this bit of work that was done by Kelly, Anthony O'Grady, Lindsay Hutley, and Derek Emus, which showed a really strong relationship between tree basal area and tree water use, we implemented this into our model, and we found that we could use this across all the tree species. So that was quite an important finding and implementation that we did to the grass model. And so, yeah, so after all the model improvements and the optimizations, we were able to get rid of those high points and get a much better fit against the daily data. And also there's a much better correlation against the monthly. So the yellow line here is the measured flux tower data. The purple line is the modeled evapotranspiration with the GRASP model before any improvements. So GRASP still does a fairly good job. So Greg's original model was still fairly good. We just improved that a bit to get rid of some of these peaks, but also every time we had these peaks, we also dried out the soil profile very quickly. So we managed to get rid of that problem and get much better fits with the model. The other thing we were able to do with this particular site, just because there was so much other data measured here, we were able to validate the different parts of the water balance against data. So we were able to get rainfall interception roughly right. Runoff and drainage was both very low in this environment, but we were able to compare that with other data sets as well. And the other really important part was the tree transpiration. So Anthony O'Grady had some measurements of tree transpiration with Sept flow loggers at this site, and we were able to compare against that as well. And the tree transpiration is actually one of the areas that we do want to improve into the future. And the last thing I wanted to show about this Alice Mulga site is how we use the satellite data. So these crosses are the measured satellite data on cloud-free days. The green line is the modeled green cover without any frost parameters. And the thing I wanted to highlight here was this is how the satellite data really helps us, because most of our measurements by the pasture scientists are done in the growing season, which is usually in summer and autumn. And so there's usually no one there out in winter measuring what happens to the grasses over winter. And if you have a look at this, where my pointer is, we were finding that the model was actually killing the grass during those frosts. And then we just found that our frost parameterization for many of these land types weren't correct, and it was only because of the satellite data that we were able to pick that up and fix it in our operational modeling. Okay, now I'll just show some of the other TERN sites. So there's not too many sites in Queensland, but I'll show some of the sites that we have started to look at. So this is flux tower sites as well as some of the Cosmos soil moisture sensors. So the first one is Virginia Park, which is near Charters Towers as well. And some people refer to this site as Weenie Creek. So the Cosmos soil moisture calls it Weenie Creek. The flux tower calls the site Virginia Park. It's on the same property. So this is one of the early sites by Ray Leunig, and there's only 3 years worth of data, but it's quite an important site for us because it's right in the heart of the grazing country, and we were keen to see how our model performs at this site. And this bottom graph shows the Cosmos soil moisture data that goes from 2011 to present. We haven't been able to match our model with the data very closely at the moment. We're just trying to get the ups and downs at this site. The other great thing about Virginia Park is Rebecca Bartley, Scott Wilkinson, Chris Stokes and others have runoff and pasture data at this site. And also it's very close to the new flux tower, Fletcher View. The second site we started to look at is the Longreach Mitchell Grass Super site, which Will spoke about. It's a QUT TERN site. It's only got 2 years of data so far. And if you can see these areas of Mitchell grass, these grasslands that are here in yellow, so that's another large chunk of Queensland that we're hoping to parameterize with the data sets from the flux tower, but also we've got a 20-year grazing trial site at Deaf with people like David Phelps. And so we're using the combination of data from David Phelps' site with the flux tower to try and improve our modeling here. And the great thing about this site so far is they've got data from a time when it was very, very dry, where there was no grass. And so we're hoping to be able to get our soil evaporation parameterization right in periods like this when it's really dry, but also when there is grass cover there to get the total ET right. And so this shows the data that they have so far for evapotranspiration and soil moisture for Longreach. We haven't modeled Robson Creek yet, but I think it'll be a really good site for us to model quickly, just to test our model to see how it works in a high rainfall, high tree basal area site. The next one is the Brigalow catchment study, where the proposed Cosmos soil moisture sensor is going up. So we're proposing for the sensor to go up in this catchment here, C3, which is the long-term pasture catchment. And this is a bit of work that we did with Ram Dalal, Craig Thornton and Di Allen on just long-term pasture biomass modeling at catchment 3. So it's a really nice long data set. It goes from 1984 all the way to present, and it's got runoff, soil moisture, recharge, and pasture biomass data. So it'll be really handy to have soil moisture from the Cosmos sites as well. And Craig Thornton is actually on the webinar today, and one of the things that Craig was proposing was to have an additional Cosmos sensor on the scrub catchment, and he's Craig's looking for some crowdfunding to get the second sensor up. So if people are interested, we could talk about that at the end of the seminar, or you could contact Craig Thornton directly. But it'd be really good to separate out the soil moisture and the management effects between the scrub and the grass catchment. Okay, and this is the Burdekin catchment study. So, this is the Wombiana grazing trial. So this is another really long-term trial that we've been working closely with the Queensland Department of Agriculture and Fisheries with, and Peter O'Regan is the main person in charge of this trial, and Peter works really closely with us, and he's on the webinar today as well. So this is data from 1998 to present, and so it's a really nice long-term data set of total standing dry matter. And there's lots of other measurements that they do at the site as well, which we use, such as grass basal area, pasture species composition, pasture degradation and recovery. And the other thing is the satellite. This is the blue line is the satellite, measured. Oh dear. The blue line is modeled green cover, and the green crosses are the observed data. So again, using the satellite data to help our modeling. Okay, and finally, the new flux tower site. So this is a JCU TERN new site that's been proposed for the Burdekin catchment. And so there's the Burdekin River there. That's just a picture of a sediment plume after the last lot of flooding in the Burdekin River. And the way we find the tower is to find the Great Basalt National Park, and it's just slightly east of there. And this is a wish list of measurements that we'd like to do at the new flux tower site. So there's a picture of Mark Silburn and I when we were installing the drainage lysimeters out at Kingsthorpe, but we won't be doing this here, but we'll try and measure recharge using some other methods. So we'll be doing the standard flux tower measurements at the site. We're really hoping to get better soil moisture at this site as well, pasture biomass and cover over the whole of the growing season so that we can use it in our pasture models. The other thing on our wish list for this site is tree transpiration with the Sept flow sensors, just so that we can get a better handle on the partitioning, recharge and groundwater monitoring. This is just a picture. This is that's the actual property, the Fletcher View Station, and there's not that many bores. That's just an image of all the bores in the region. There's not many, and so we thought it'd be really great to get a groundwater bore at this site, just to help us with our water balance modelling and to close that gap with the deep drainage and recharge. This is a soils map of the region. So when we first heard about the flux towers at this site, we were a bit concerned about whether it was going to be representative for the whole of Queensland. And luckily I got some help with soils from the soil scientists at DES and DNRME, so Jim Payne, Tessa Chamberlain and Matt Crawford. And we worked out that this red soil here called the Hillgrove was fairly representative. So it's a red basalt land type, a red ferrosol soil on basalt, very similar to the soils here in Toowoomba, and it was quite representative. So that's where we're hoping to locate the flux towers. This is a vegetation height map of the area. So Lindsay Hutley was helping me work out how many Sept flow sensors we were going to need, and we didn't even know how tall the trees were at the site because we haven't actually been on the site yet. And so we were scratching our heads, working out, trying to work out the height, and then Peter Scarp's name came up. And later that night, I sent Peter an email, and an hour later, I got a map telling me how tall the vegetation at this site was with his product, and he cross-checked that against ISET as well. So the vegetation is about 10 to 13 meters at this site. And so we've been getting quotes for things like subflow sensors and everything else we'll need at this site. And the last thing is this idea that we had of having paired flux towers. So Dennis Badolci has been talking to us ever since the OSFlux conference from a few weeks ago, helping us with some guidance on the main flux tower that we already have funding for. And one of the things that Dennis suggested was getting a second tower to put over the grasses, so just so that we could separate out the fluxes between the tree canopy and the understory of the grasses. And so what we're doing at the moment now is trying to get some funding for the second tower. And so this is just a map of all the different stations and sites at this location, very close to Charters Towers in Townsville, where James Cook Uni is. So this is the Fletcher View Research Station. This is the Wombiana grazing trail, where I showed the results from. That's the Virginia Park Weenie Creek site, and another grazing trail that DAF has called the Spyglass Research Station. So there's been a lot of interest in this area, and I think it'll be a really good site for collaboration with lots of different groups of people. And so in conclusion, yeah, we improved the modeling of evapotranspiration using the TERN flux tower data, and we're very keen to have more sites in Queensland to help us with this. We were able to improve the parameterization for the mulga woodland savannas for Queensland and Australia as well. And the bonus was improved tree transpiration for all woody vegetation in Queensland and Australia. We demonstrated with this bit of work how satellite-derived vegetation cover can be used to enhance the modeling of the grass and the woodland systems. And finally, we're very excited about the greater collaboration with TERN, the OSFlux community, and James Cook Uni with our work in Queensland. And thank you. Thanks, Angela.
That's great. Thanks, Joe. So I guess what we'll do now is I'll share my screen again. Okay, so we've got time for a few questions, except I can't, oh yes I can, here they are. So a question for Joe. Will showed that there are 12 to 15 SuperSites in Australia. Is the data coming from these and the other TERN sites providing enough granularity for modelers, or is there too little real-time data to allow you to reduce uncertainty in your models?
So as a model, I'll say that there's never enough sites, and certainly in Queensland there hasn't been enough sites. That's why we've had to go through and pull out data from places like Virginia Park, and we were able to go across the border to our neighbours in the Northern Territory, and because the soils over the mulga lands are usually red canter soils, and we knew that the climate, the soils and the vegetation was the same, we were able to go across and pull that across and use it for Queensland. But you know, I just don't think there's enough sites to, and we're really glad to have a few more sites.
That's a very good answer, Joe. Never enough data. Okay, and so a question from Jason Berenger. Thanks, Joe. Good stuff. I wonder if you could comment on what data is most important for your work and whether we're collecting everything you need. Are there any big gaps in information that would be important to you?
That's a great question. Jason, you should never ask a modeler that, because I can send you a few pages of all the data sets that I need. One of the things that would be really useful though is, which we found was a lack, was we found that at some of the sites people weren't even measuring tree density. And so we had to do a fair bit of trying to work out what the tree density was ourselves, just so that we could use the data. So that's our first one, is can everyone measure tree density at their sites? The other one is in the understory. It would be great if we could have a few measurements over the growing season, so we knew what the pasture biomass was over the growing season. For soil moisture, it'd be great if there was some gravimetric soil moisture to cross-check the sensors. But yeah, I've got a really long list of things that we could do, and hopefully we can do that, a lot of that at Fletcherview with all our partners.
Well, it's always good to have lists ready, isn't it?
Yeah.
Matthew Webb asking you, Joe, what crowdfunding platforms are you considering using to fund the additional towers?
Well, at the moment, I haven't resorted to crowdfunding yet. Although I did do a talk for National Science Week at my daughter's school, and it was really funny. At the end of the talk, the kids were interviewed by the local TV, and the kids could remember how much the towers cost and how tall they were. So I might take a bucket around, try and collect some money. But at the moment, I'm trying to get funding through our university because we've got equipment funding, and also there's a few other people with big projects that are interested in this. So I'll go with them first. But if all else fails, I might have to look at crowdfunding. Craig Thornton's also really interested in crowdfunding his second Cosmos sensor for soil moisture, and that costs $20,000. And I think Craig's already got 5,000, and he's looking to get money from other partners because a lot of people use the Brigalow catchment data set.
A question here from Elise Pendle. Great presentation, Joe. Do you have suggestions for how to separate grazing effects from climate effects on green cover?
Oh, that's a really good question, Elise. The only way we do it is with the modeling and the data. Yeah, I can't quite think of a way to separate out the management from the climate. Our group has a lot of history with working with climate and especially climate variability. And yeah, sometimes it's hard to separate out the signals between the two of them.
And a comment from Beryl Morris. Well done, Will and Joe. It's fantastic to see how national research infrastructure can provide a service to researchers like Joe, and how the research can be applied to land management questions. So
Thanks,
that's
Sarah.
about all the questions that we had in the Q&A. So beyond that, thank you so much, both Joe and Will, for participating and giving us a really great insight into what's happening in ecosystem processes. Our next seminar will be held on the 4th of November, and that's Standardising Environmental Information Collection, Harmonising Australia's Approaches. Again, a panel discussion, this time led by Associate Professor Ben Sparrow, who's leader of the TERN surveillance platform and based at University of Adelaide. And I think that's us done for the day. So thank you once again for participating, and stay safe and well, everybody. Bye.
Thanks, Angela.
Thanks very much.
Thanks, everyone.